Thank you so much for the introduction. I just want to start by saying I had an amazing time, learning about maize this summer. And so I tried to kind of sprinkle in photos of maize where I could. So I'll kind of walk you through those as well. But this is maize growing in the BTI greenhouse. So first things first, a quick little outline. So I'm going to start with some background on the project and then talk about the experimental goals and then work through the methods I was using to address those goals, in addition to talking about results, a quick summary of what I learned, and then some future directions and acknowledgements. And this is maize growing in a growth chamber at BTI. So first, most importantly, a little bit of background on rubisco. It's the most abundant protein in leaves, and it catalyzes assimilation of inorganic carbon into essentially sugars. So this serves as basically the foundation of kind of our biosphere in terms of being a primary producer. And it's actually quite complicated and large enzyme with multiple subunits. So it has a variety of assembly factors as well as a rubisco activase, and that's necessary for putting both of these two different subunits together into one large hollow enzyme. So as you can see here, you've got in red circles the large subunit and the small subunit, and there's about 8 of each, and they assemble into this one massive enzyme. And then this is RAF1, which is an assembly factor, and I'll come back to that later. But as important as rubisco is, it actually has some inefficiencies, as you may or may not have heard about. What it's supposed to do for the plant is take in carbon dioxide gas and then funnel it into the Calvin cycle, where you can produce useful things like NADH and ATP. But unfortunately, if the concentration of oxygen is too high next to rubisco, it will actually perform photorespiration instead. And this produces a different product. And though it can be recycled, it's actually pretty metabolically wasteful and expensive. So it's less than ideal as a situation. So as part of this, rubisco has actually adopted, plants have evolved different ways to address this problem of photorespiration. And maize, for example, practices C4 cycle. And essentially the principle of C4 photosynthesis is that you can separate rubisco from gas exchange via a membrane. And this allows the plant to control the quantity of carbon that's near rubisco, basically. So if you have rubisco over here in the bundle sheath cell, you have this other molecule separated that acts as a carbon dioxide acceptor called PEP. And significantly, pay attention to this for later, is the enzyme PPDK, which is in charge of regenerating the molecule PEP. And so basically, the cell is able to take in carbon dioxide and oxygen over in the mesophyll, perform chemical reactions, and just transport carbon into the bundle sheath. And that's where it can hand it off to rubisco and kind of prevent photorespiration from occurring. So if we're thinking about trying to improve photosynthesis, the question is what has been done in the past? So the Stern lab was able to create a maize line that's overexpressing the large and small subunits, which I pointed out earlier, in addition to RAF1, which is the rubisco assembly factor 1, which I also pointed out in that assembly diagram. And there were some really promising results because they were actually able to show that those maize overexpressing those proteins had increased carbon assimilation and, interestingly, also increased resistance to chilling stress. So here on the slide, you see what's called an Aci curve. And I ran some of these as well. So I'm just going to explain the axes while I'm on this slide. So on the X slide, or on the X axis, basically use a machine called a Lycor to change the internal concentration of carbon within the cell. And then you can measure how much carbon is being assimilated. And that's what you see on the Y axis. So basically, you can kind of see how as carbon increases in the cell, the amount of carbon being assimilated also increases until you reach a sort of saturation point. So here in A, where there was no chilling stress on the maize, you can see a kind of saturation point over here, maybe around 35, 40. But down here, when the maize was exposed to chilling stress, that saturation point is a lot lower. One other important trend to notice is that these white empty boxes are the maize that are overexpressing RAF1 and LSSS, and those tend to be a little bit higher on the curve on both of these. And so that's how we assessed that they were assimilating more carbon and were more resistant to chilling stress. So I might have mentioned in the title slide this grass called Miscanthus Gigantius, and it really is, it's a gigantic grass. It also performs C4 photosynthesis. It's a perennial, so it captures more light earlier in the season. And it also has a higher biomass produced than maize. So it's just kind of a cool plant in general. If you can see, it's just massive and tall. But the trait that's more interesting to us is that it's actually pretty chilling resistant. So in a previous study, researchers took PPTK that had been isolated from Miscanthus that had either been chilled or not chilled, and then they performed basically enzyme curves to look at the activity of those enzymes. And as you can see in maize, the PPTK that came from a chilled plant had a much lower maximum velocity of action than did the PPTK that was isolated from chilled plants in Miscanthus. So you can kind of see that divergence on the maize graph that you don't see in Miscanthus graph. So this brings us to the experimental question for my summer. Basically, if we overexpress PPTK from Miscanthus in maize, could we improve carbon assimilation and could we improve chilling resistance in this maize? So basically, we're looking for a twofold effect. One, we're increasing PPTK. So hopefully we'll see more flux through that pathway, because back in that original paper I was showing, while we did see increased carbon assimilation, we didn't see nearly as much as we would expect based on how much Rubisco was overexpressing. So that made us think maybe we should be looking for another limiting step in that C4 pathway. So here, if we're with PPTK, not only are we having more PPTK to funnel more carbon to Rubisco, we're also potentially expressing a PPTK that's more chilling resistant and would make that pathway more effective at colder temperatures. So the idea is that if Rubisco is in the bundle sheath and we're overexpressing PPTK, then theoretically we should be able to improve C4 pathway flux. And that's basically the hypothesis. If we're overexpressing RAF1 LSSS, the assembly factor, in addition to Rubisco and PPTK, maybe we can continue to just kind of bump up the C4 efficiency to where it could be based on how much Rubisco we had in the plant. So in order to address this question, I used a few different methods. One is immunoblotting. And basically the idea is that we can detect a protein of choice with a primary antibody and then detect that primary antibody with a secondary antibody. And the secondary antibody has a signal that allows us to visualize its binding. So that's the little yellow star. I also took maize phenotype measurements, so height and then leaf length and leaf width and things like that, in addition to performing chilling stress experiments, so putting the maize in colder temperatures. And then, as I mentioned, you can get Aci curves using a machine called a Lycor. And so I also performed photosynthesis measurements. So my chilling stress protocol, I started with plants that had just been sprouting in the greenhouse, essentially, and I was able to take photosynthesis measurements at that point as a pre-stress measurement. I then put them in a cold chamber with 12-degree nights, 14-degree days for 4 days. And then the morning of taking my post-stress measurements, I put them in a cold room, so 4 degrees C, just to make sure they really were really cold, and then took photosynthesis measurements again. And this is actually a shorter timeline than the Stern lab typically uses for chilling stress, just because I was on a little bit of an intern timeline as well. And that's why I added the cold room step. Actually, typically the Stern lab does a longer period in just the cold growth chamber. But I was able to confirm that my plants did, in fact, experience chilling stress, because you can see this buildup of anthocyanin and pigment in the leaves, which is beautiful. It means the plant is not happy, but but it looks awesome. So what were my results? First off, I thought it would be important to discuss how I figured out the genotypes of the plants I was using in this chillings trial, because in plants, you can't necessarily confirm that what you crossed with originally ends up actually being expressed in the plant itself. So I used Western blotting. So here you can see that I had 3 constructs I was hoping to see, which was 4 different proteins I was hoping to overexpress. So the PPTK was tagged with flag so that I could visualize it using an anti-flag antibody. And that should be about 100. So anything on this 100 line that looks big and thick and bright is overexpressing PPTK. And anything down here, my Rubisco subunits are in 1 construct with 1 tag. So I was able to screen for them both using the same antibody. And that should be about the 50. So any of these big bulky lines are overexpressing the Rubisco subunits. And then here you can see I screened for RAF1, which is the assembly factor, and that should be about 75. So as you can see, I only had 2 plants that were overexpressing all 4 proteins, unfortunately. So n equals 2 is a small sample size, but I kind of went forward with the experiment. And this is the same method I used to find plants that were just overexpressing RAF1 LSSS and just overexpressing PPDK, and those served as controls in my experiment as well. So first off is phenotype difference. And this was kind of glaring just as I was looking at my plants. There was a pretty visible height difference. So the RAF1 LSSS, which was previously studied in the lab, continued to be taller and outperform the untransformed control, which is called high 2. And interestingly enough, the plants overexpressing PPDK, both alone and with the other constructs, seem to be a little bit shorter. And just here you can see that the plants with PPDK just look a little bit shorter than the plants that aren't overexpressing PPDK. And then I began my photosynthesis measurements. And unfortunately, a lot of the Aci curves didn't necessarily show a huge difference. So I kind of put one up there so you can see what they look like. And so that red pre-stress curve has that higher amount of assimilation, and then it does bump down and have lower assimilation post-chilling stress. And this is actually one of my quadruple overexpressors, but there wasn't a huge difference in the appearance of these plots between them. So I also graphed the averages because I had 3 replicates for most of my genotypes and then 2 for my 4 overexpressing. And you can see that the gray PPDK tends to be a little bit lower on this graph, and the RAF1 LSSS, so the orange, tends to be a little bit higher. But overall, they look pretty similar, and you can see that they all seem to respond to chilling stress at like pretty similar levels. So I would need more replicates to prove that there was any difference happening in that response to chilling stress. So overall, I did see phenotypic differences in my different overexpressing transgenics, and I was able to successfully induce chilling stress. However, PPDK might not be the limiting step that we were hoping to find in terms of what's slowing us from improving C4 flux even more. And that could be for a couple different reasons. It's possible that PPDK itself is just bad for the plant, and we didn't know that before. And the negative effect it does have on the plant could be masking the aid that it's giving in the cold response. It's also possible that the PPDK we are overexpressing is from a different species. And so the plant might not be either expressing it or responding to it as we would hope it would. And so further experimentation will definitely have to come into play there as we're trying to evaluate why we didn't get the results that we expected. And this is corn silks that have been freshly pollinated. So you can see a little bit of the dried anthers on there as well. And then last is kind of where we want to take this project in the future. So I feel that we definitely need to increase the scale of the experiment. We could increase the chilling period. We could increase the sample size of the plants, which would kind of allow for statistics to be a little bit more effective. We could also investigate other C4 pathway proteins to try and find that elusive limiting step that we kind of ran across in previous experiments. And we can also look into other C4 species that are environmentally resilient, like Miscanthus, to try and mine those pathways for more resilient proteins as well. And these are called the brace roots, and sometimes they have a really gorgeous magenta color. So just want to end with some acknowledgements. Thank you so much to the Stern Lab for taking me in. Elena was an amazing mentor, but I learned so much from everyone else in the lab as well. And then thank you to everyone who made the REU program possible, in addition to the greenhouse staff and the stockroom staff who took care of my maize for me when I wasn't taking care of it. So, and then as a side note, you didn't see a lot of them in the presentation, but I ran 44 Western blots this summer. So shout out to my blots. Thank you. We have
time for a few questions.
Yeah, absolutely. I took measurements of the length of the newest fully expanded leaf and width measurements for that as well. And I use that to calculate a leaf area. But in the graphs that I was making and kind of the calculations I was doing, I wasn't seeing any difference at all. And they were kind of just like large standard deviations and things like that. So I chose not to show them on the presentation. So it's not necessarily known the mechanism. Based on that paper, the fact that even after it was taken out of the chilling stressed plant, it still performed more poorly at the same temperature as the non-chilled protein suggests that it's some sort of like stability. So maybe there's an extra covalent bond or something that's broken in response to chilling stress. So it's kind of permanently maimed, I guess, after it gets chilled. And then I believe it's not, it definitely has a, I want to say tetramer. Yeah. So it could potentially be an assembly problem as well, like trying to express a foreign protein in maize. Yeah, absolutely. Yeah. Yeah, absolutely. There's a different project that I kind of helped out with that was overexpressing RAF2, which is a different assembly factor. And then my mentor is also looking into overexpressing a protein from agave, which might be heat stress resistant. And that's also part of the C4 pathway. Thank you.